Sushruta for NEET is now available on the Google Play Store. Try it out for free.
In the last class, we have studied about various aspects of plant growth and development. We have understood what is exactly the growth, what is the development, and what all required for the growth and development. And in today's class, we are continuing the same aspect. One of the most important terms with regard to the growth and development in plants are differentiation, de-differentiation, and re-differentiation. Always, students get confused between these three terms. If you follow the words verbatim, you'll understand these three terms very clearly, and later you find it very easy. Let us study that. The cells derived from root apical and shoot apical meristems and cambium differentiate and mature to perform specific functions. We have seen yesterday. Root has actively dividing growing region called root apical meristem. Shoot has shoot apical meristem. The meristem is the area where the cells actively divide. Once these cells are formed, either from the root apical meristem or shoot apical meristem, they start differentiating. Once they start differentiating, they start doing a specific function. And these tissues, which are differentiated and start doing different function, we call them permanent tissues. You know, permanent tissues are of simple type or complex type. In the same way, the cambium which is present between the xylem and phloem in the vascular bundles also shows a division, or it also shows a dividing. It also shows a division of the cells, or it also shows more number of cells resulting from the division. And these cells also get differentiated into one or the other structure with the specific function. So this is what you have already seen in the previous classes. This act leading to maturation is termed differentiation. This act of the cells which result from the division of root apical meristem or shoot apical meristem, or which result from the division at the cambium, when these cells which are formed newly, they mature to take up a particular function, and this is what is called differentiation. During differentiation, cells undergo few to major structural changes. Whatever the cells are formed from the root apical meristem or shoot apical meristem or cambium, once these cells are formed, these cells undergo few to major structural changes, both with regard to their cell walls and protoplasm. Right? So this is what you have. For example, to form a trachea element, the cells would lose their protoplasm. So you see here vessels, tracheal vessels. In the tracheate or in the vessels, xylem vessels. If the protoplasm is there inside, how the water with the minerals can move? It is not possible. So, these which are formed as a result of differentiation, these should undergo certain structural changes, maybe simple change or complex change, so as to enable these newly formed cells which result in tissues to take up their function. As I said, for example, to form a trachea element, the cells would lose their protoplasm. Once the protoplasm is lost, they become empty now. Once they are empty, there is scope for the water to movement through this empty area. You know what are the tracheids? Tracheids are elongated cells in xylem of vascular plants that serve in the transport of water and mineral source. Tracheids are one of the two types of tracheal elements, vessel elements being the other. You know, in the xylem, if you see, there are four elements: xylem vessels, tracheids, xylem parenchyma, and the xylem fibers. So this is what you see here. Tracheids, unlike vessel elements, do not have perforation plates. Tracheids do not have, here you can see perforation plates. It is a vessel. But here, there are no perforation plates. So what I wanted to tell, once these xylem vessels or tracheids are formed, these are formed because of the division, what you see inside the cambium. Once these are formed, they have to take up a specific function. In order to take up the specific function, they should undergo certain modification, maybe simple or complex. They also develop a very strong, elastic, lignocellulosic secondary cell wall. Lignocellulosic means made up of lignin and cellulose. They also develop a very strong, elastic, lignocellulosic secondary wall to carry water to long distances even under extreme tension. Many times, you may be knowing, the trees grow to the extent of 200 to 300 feet. 200 to 300 feet. If the water has to move from the soil up to 200 to 300 feet, the vessel which it takes this water must be strong enough. The tracheids must be strong enough. Therefore, the these vessels and tracheids develop a very strong, elastic, lignocellulosic secondary cell walls. Right? Now, of course, with your previous study, try to correlate the various anatomical features of anatomical features to encounter in plants. You encounter in plants to the functions they perform. That means, try to recall what the xylem is made up of, what the phloem is made up of. Right? In the phloem, we come across sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Imagine the structure of them. And they're all specialized, modified to do their own function, their destinated function. Plants show another interesting phenomenon. Now we have seen what is differentiation. During differentiation, the cells formed from the cambium or that of the root apical meristem or shoot apical meristem, they get differentiated to perform a specific function. Structurally, they are differentiated. Structurally, they are modified to perform a given function. Now, let us see another interesting phenomenon. The living differentiated cells, what we have already seen, the living differentiated cells that by now have lost the capacity to divide, once Once this root apical meristem or shoot apical meristem or cambium divides and forms some structure, when they undergo modification, when they take up a particular function, immediately they lose the capacity to divide further. Once they are modified, once they take up a particular function, immediately they lose the capacity to divide further. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. Whatever the cells are formed, whatever the cells got modified, whatever the functions they have taken up, whatever these structures which are formed as a result of differentiation. What I said, the structures which are formed as a result of differentiation, they lose the capacity to divide, but again they get the capacity to divide. That is what is called de-differentiation. This is called de-differentiation. This phenomenon is termed as de-differentiation. For example, formation of meristems. Formation of meristems, interfascicular cambium. Interfascicular cambium is in between the two vascular bundles. And cork cambium from fully differentiated parenchyma cells. So, formation of meristems, interfascicular, in between vascular bundles, and cork cambium, which you see on the outermost part of the tree, from fully differentiated parenchyma cells is examples for de-differentiation. While doing so, such meristems, all tissues are able to divide and produce cells that once again lose the capacity to divide, but mature to perform specific function and get redifferentiated. So, I repeat, once the differentiated cells get the ability to divide, it is called de-differentiation. Once these cells which got the ability to divide by de-differentiation, after forming the cells, again they lose the ability to divide. After forming a specific structure, after getting specific function. This is what is called redifferentiation. Don't get confused. Differentiation, de-differentiation, redifferentiation. So, I again repeat the phenomenon of differentiated cells which had lost the power to divide. Getting the power again to divide is called de-differentiation. The examples for de-differentiated tissues are interfascicular cambium or that cork cambium. Once these are formed, now these are formed as a result of de-differentiation. Once these are formed, in fact, they have to lose the ability to divide. By doing so, such meristems or tissues are able to divide and produce cells that once again lose their capacity to divide, but mature to perform the specific function, that is redifferentiation. Let us see the examples, then you will understand better. Differentiation, de-differentiation, redifferentiation, once again. Transformation of derivatives of primary meristem into permanent cells is a differentiation. Transformation of derivatives of primary meristem. Root apical meristem, shoot apical meristem, these are the primary meristems. Into permanent cells is a differentiation. Regaining the meristematic activity by permanent living cells is called de-differentiation. Once the cells are differentiated, they lose the ability to divide. Again, they get the ability to divide. That is the de-differentiation. Transformation of the derivatives of second meristem into permanent cells is called redifferentiation. Again, let us see the examples for this. See here. Root, apical, shoot, apical meristems and cambium differentiate and mature to perform specific function differentiation. Clear? It is very clear. Root apical meristem, shoot apical meristem, and cambium differentiate and mature to perform specific function differentiation. Please have the screenshot of this. Always it is going to help you. Cell wall and protoplasm undergoes changes. Differentiated trachyarrangement cells lose their protoplasm, develop a very strong elastic lignolusing secondary cell walls and all. These are all products of differentiation. And these are all have lost the ability to divide. But what happens? The living differentiated cells, these all living differentiated cells that by now have lost the capacity to divide, can regain the capacity of division under certain condition. This is called de-differentiation. These which have lost the ability to divide, they gain the ability to divide. That is called de-differentiation. Development of interfascicular cambium and cork cambium from fully differentiated parenchymal cells is the example of de-differentiation. Right? These are the examples of de-differentiation. Right? As a result of de-differentiation, as a result of getting ability to divide again, they form this. Once these are formed, what should happen? These should also lose the ability to divide. Cells produced by the de-differentiated tissue again lose the capacity to divide and mature to perform specific function is called redifferentiation. I hope it is clear. Differentiation, de-differentiation, and redifferentiation. Very, very important concept. Let us look at the examples for that. A meristem at the tip of the plant shoot or root that produces oxygen and causes the shoot or root to increase in length. Growth that originates in the apical meristem is called primary growth. Cambium, a layer of delicate meristematic tissue between inner bark or phloem and wood or in the wood or in the wood or xylem is the cambium, which produces new phloem on the outer side and a new xylem on the inside in stems, roots, etc., originating all secondary growth in plants and forming the annual rings of wood. Tracheids are elongated cells in xylem of vascular plants that serve in the transport of water and mineral source. Interfascicular cambium is to bring about secondary growth in a dicot stem and the root. Cork cambium is a tissue found in many vascular plants as part of epidermis. The cork cambium is a lateral meristem and is responsible for secondary growth, that is increase in girth, that replaces the epidermis in roots and stems. It is found in woody and many herbaceous dicots, gymnosperms, and some monocots. So this is a little bit more explanation with regard to the structures that are formed. Now, question. List some of the tissues in a woody dichotomy plant that are the products of redifferentiation. Differentiation, de-differentiation, redifferentiation means finer structures that are formed which take a particular function. Second xylem, second phloem, phellum, phelloderm, etc. are the products of redifferentiation. Right? Then, how would you describe a tumor? Plant tumors, aggregates of cells that have multiplied excessively, are usually caused by a bacterium, virus, fungus, or may develop as a result of structural damage. It is not concerned with anything else. What would you call the parenchymal cells that are made to divide under controlled laboratory conditions during plant tissue culture? Parenchymal cells that are made to divide to form callus under controlled laboratory conditions are examples of de-differentiated tissue. Differentiated, de-differentiated tissue, right? Now you have to know a little bit of what is meant by indeterminate and determinate. Definition of indeterminate growth. Indeterminate plant growth in which the main stem continues to elongate indefinitely without being limited by terminal influences or other reproductive structures called indeterminate. Continuous growth, not determinate, indeterminate. Growth characterized by sequential flowering from the lateral or basal buds to the central or uppermost buds. Definition of determinate growth. Plant growth in which the main stem ends in an inflorescence or other reproductive structure and stops continuing to elongate indefinitely with only branches from the main stem having further and similarly restricted growth. Growth characterized by sequential flowering from the central or uppermost bud to the lateral or basal buds. So just remember only one thing, indeterminate growth means continuous growth. Determinate growth means the height of the plant stops with a flower on the top, no more further growth in that. It is determined. Now we may say that even differentiation in plant is open because cells and tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of a cell or tissue is also determined by the location of the cell within, where it is located. For example, cells positioned away from the root apical meristem differentiate as root cap cells while those pushed to the periphery mature as epidermis. Those which are there in the state nearer to that, they differentiate as root capsules. Those which are present at the sides, they differentiate at epidermis. Can you add a few more examples of open differentiation correlating the position of a cell to its position in an organ? It is possible. Now let us look into another important concept, what is called development. Development is a term that includes all changes that an organism goes through during its life cycle from germination of the seed to senescence. What is development? Development is a continuous process, right from birth to death, right from germination of the seed to senescence, that is becoming old and falling down, getting aged. So that is development. Generally, in case of animals and all, growth stops at a particular age, but the development continues. In case of the plants, both growth and development is continuous. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given here. It is also applicable to tissues or organs. You see, meristematic cells. Meristematic cells, they do show plasmatic growth, and later they get differentiated. During that, you come across elongation. Then they get mature, they form mature cell, and this mature cell undergoes later senescence, resulting in the death. So these are the sequence of development process in a plant cell, right? The same thing I have learned here. Growth due to repeated divisions of formed cells is called plasmatic growth. Growth due to repeated divisions of the formed cells is called plasmatic growth. One cell becomes 2, 4, 8, 16, 32, which results in increase in length or increase in girth. Once the cells result from the meristem cells, first they get elongated, cells get elongated, they get differentiated, and we say that they are mature, they take up a particular function, they get matured, and after maturing, they have to die once they get aged, right? Plants follow different pathways in response to environment or phases of life to form different kinds of structure. That means plants follow different pathways in response to environment or phases of life to form different kinds of structure. You might have seen some of the plants. They look slightly different in winter and different in summer. Or they look differently when they are under the water and totally different when some of the leaves are present above the water. This ability is called plasticity. That is, heterophyly in cotton, coriander, and larkspur are examples for that. Not in all, not in every plant. You see that some plants, they look different when they are partially covered with water and partially they are exposed to the air. This ability to show different kinds of structures under the influence of environment as a response to environment is called plasticity. The plasticity is well observed in cotton, coriander, and larkspur. In such plants, the leaves of the juvenile plant are different in shape from those in mature plant. See here, juvenile plant leaves are different from adult. Here, the plants which are there in terrestrial habitat, the same plant is different when it is in water habitat. So this is what is called heterophily in case of a larkspur and in case of a buttercup, and similar change you see even in our coriander leaves, the one which we use in our kitchen. See, heterophily in coriander, also known as cilantro. Look how big these leaves are because these are grown above the ground, and these the same coriander leaves when they are with more water like a sewage water and all, then you can see that the leaf structure is different. Under the environmental condition, they do show different structures in the leaves, and this is the best example for the plasticity. On the other hand, differences in shapes of leaves produced in air and those produced in water in buttercup also represent heterophyllous development due to environment. This is in buttercup. This phenomenon of heterophyly is an example of plasticity. Ability of plants to follow different pathways to form different structures of it in response to environment and phase of growth is called plasticity. Already have seen heterophyll in cotton, larkspur, coriander, and ranunculus. The shape of adult leaves is different from juvenile leaves. Right? Thus, growth, differentiation, and development are very closely related events in the life of a plant. Growth, differentiation, and development. Broadly, development is considered as the sum of growth and differentiation. Development is sum of growth and differentiation. Development in plants, both growth and differentiation, is under the control of intrinsic and extrinsic factors. As we have studied in the last class, intrinsic factors are internal, extrinsic factors are external. The intrinsic factors include both intracellular, that is genetical, or intercellular chemicals such as a plant growth regulator, while the latter include light, temperature, water, oxygen, nutrition. These are extrinsic factors. External factors. External factors include light, temperature, water, oxygen, nutrition. Intrinsic factors are the factors which are present within. Within means within the nucleus. Intracellular means genetic, within nucleus. Intercellular means in between the cells, that is chemical. Such chemicals are called plant growth regulators or plant hormones. We are going to study the plant growth regulators now. The plant growth regulators, PGR, we generally call that PGR, are small, simple molecules of diverse chemical composition. Plant growth regulators, simply they are hormones, plant hormones. Just like we have animal hormones, here the plant hormones. They could be indole compounds, indole 3-acetic acid, IAA, indole 3-acetic acid. You must know this. It's very important, always ask it. They could be indole compounds, adenine derivatives like N6-perfluoryl aminopurine, kinetin, derivatives of carotenoids, abscisic acid ABA, abscisic acid is called ABA, abscisic acid, terpenes, gibberellic acid GA3, or gases ethylene C2H4. So these are all the different compounds. Indole compounds you must know, which are indole compounds, which are adenine derivatives, which are derivatives of carotenoids, or which are gases. Plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones. All these are one the same. We call plant growth regulators by plant growth substances name, or plant hormones, or simply they are called phytohormones. The plant growth regulators can be broadly classified into two groups based on their function in a living plant body, whether they promote growth or whether they inhibit growth. Are they going to promote the growth or are they going to inhibit the growth? Based on that, plant growth regulators are of two types. One group of plant growth regulators are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, trophic growth, flowering, fruiting, seed formation. These are all growth promoting activities. Activities like cell division, it is a growth. It is considered as growth. Cell enlargement, pattern formation, trophic growth, flowering, fruiting, seed formation. These are all the events of promoting growth. These are also called plant growth promoters. That is, you must know plant growth promoters are three: auxins, gibberellins, cytokinins. Always you must know which are the plant growth promoters and which are the plant growth inhibitors. These are all plant growth promoters. Right? So, the plant growth regulators of the other type play an important role in plant responses to wounds and stresses of abiotic and abiotic origin. So, here you have seen plant growth promoters. These are plant growth promoters. But we have another category, plant growth regulators. Plant growth regulators are of two types: plant growth promoters, plant growth inhibitors. The plant growth regulators of the other group, that means inhibitors. play an important role in a plant response to wounds and stresses of biotic and abiotic origin. Biotic means stress caused by living organisms. Abiotic means stress caused by non-living things. They are also involved in various growth inhibiting activities such as dormancy and abscission. They are involved, these another type of growth regulators are also involved in various growth inhibiting activities such as dormancy, remaining in a less active condition, and abscission. Abscission. What is this abscission? Abscission, the natural detachment of parts of a plant, typically dead leaves and ripe fruits. Abscission means falling of the fruits, falling of the leaves. So these plant growth regulators of another type are involved in various growth inhibiting activities such as dormancy and abscission, making the fruit to fall, making the leaves to fall. Abscission is a natural detachment of the parts of the plant, typically dead leaves and dry fruits. Dead leaves and dry fruits, they have to fall. It is abscission, it is natural phenomena. The PGR, abscisic acid, belongs to this group. So you have PGR, abscisic acid. What is that PGR? Plant growth regulators like abscisic acid belongs to the group of inhibitor. The gaseous PGR, ethylene, gaseous plant growth regulator, ethylene, could fit either of the groups, but it is largely an inhibitor of growth activities. Ethylene can be fit into growth promoter also, but it also plays a growth as a growth inhibitor, but mostly ethylene, gaseous form of plant growth regulator, is largely an inhibitor of growth activity rather than promoter of growth. Now let us study these in detail. It is simply knowing, and there is no other go. You have to simply memorize. There is no other go. There is no other easy way to remember this. Up to now, it is fine. Very nice. You can understand each and every point. But now we are looking into the different types of plant growth regulators. But now, before that, we shall look into the discovery of plant growth regulators. Interestingly, the discovery of each of the five major groups of plant growth regulators. What are the five major types? You know, three are growth promoters: auxin, gibberellins, cytokinin. Auxins, gibberellins, cytokinins are growth promoters. Two are growth inhibitors, namely the abscisic acid and ethylene. These are the growth inhibitors. All these five got discovered accidentally. It was not they got discovered purposefully, with specific efforts. No, they all got discovered accidentally. All this started with the observation of Charles Darwin. Yeah, same Charles Darwin, father of evolution. The all this started with the observation of Charles Darwin and his son Francis Darwin, when they observed that the coleoptiles of canary grass, coleoptile is the part which is supposed to form the shoot. Coleoptiles of canary grass responded to unilateral illumination by growing towards the light source, phototropism. Whenever these canary grasses coleoptiles were growing, when they focused the light from one particular area, one particular direction, the coleoptiles which were going, they started bending and moving towards the source of light. Of course, today you know what is there, sir? It is simple, everyone knows it. But at that time, it was something different. It was really great observation. Canary grass is a plant, Phalaris canariensis, belonging to the family Poaceae. You know, all the grass belong to the Poaceae. Originally a native of the Mediterranean region, it is now grown commercially in several parts of the world for bird seed. It is grown to get a bird seed. What is a bird seed? Bird seed, a mixture of seeds, as hemp, millet, and sunflower, used for feeding caged and wild birds. It is a bird feed, bird seed, bird feed. Okay, and it's mainly obtained from that of the canary grass. Right? After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. See, this is what I said. The coleoptile, which was supposed to form the shoot, yeah, it is going to form the shoot. When the light was given in a particular direction, it started bending towards the source of light, right? Experiment used to demonstrate that the tip of the collectile is the source of oxygen. After that, they wanted to see what is that, what is there in that particular tip, why that, what made the tip of the collector to bend towards the source supply. When they started looking into what chemicals are present here, the chemicals found was nothing but oxygen. So, oxygen was isolated by FW went from tips of collectors of oat seedlings. You might have heard about oatmeal, right? And oat seedlings. So, from the collectile of oat seedling, oxygen was isolated by FW went. It is very important. Always ask a question: who isolated? Who invented these different types of phytohormones? Oxygen was isolated by FW went. Same thing. The Bekene, foolish seedling. It is commonly called foolish seedling. This is Bekene. The Bekene disease of rice seedlings was caused by a fungal pathogen Gibberellica fugicari. So there is a disease called Bekene. The Bekene disease of rice seedling is the normal. This is the Bekene affected rice seedling was caused by a fungal pathogen Gibberella fugicari. It was E. Kurosawa reported the appearance of symptoms of disease in the rice seedlings when they were treated with the sterile filtrates of the fungus. Sterile filtrate fungus. This is the fungus. When they applied this fungus on this particular healthy rice seedlings, they developed this particular disease, Bekene disease. So the active substances were later identified as gibberellic acid. So what it contained when they took the fungal extract and this sprayed this fungal extract or sterile filtrate of the fungus, sterile filtrate of the fungus onto the healthy plants. They developed this Bekene disease and they wanted to find out what is there in this fungal extract, fungal, and this sterile filtrate of this fungus. When this sterile filtrate, that means filtrate is obtained after the filtering this the fungal solution, and the active substances were later identified as gibberellic acid because it was obtained from Gibberella. What is the importance of this gibberellic acid? F. Skoog and his co-workers observed that from the internodal segments of tobacco stem, internodal segments of this tobacco stem, the callus proliferated only if, in addition to axin, the nutrient medium was supplemented with one of the following extracts of vascular tissues, yeast extract, coconut milk, or DNA. So this is another one. We will be coming to all these different types of the phytohormones one by one later. So I have said about first how oxygen was isolated. Now I said how that this gibberellic acid got isolated. Now it is a third one. So you must know gibberellic acid is due to Kurosawa. Now you must know Yafusko. And his co-workers observed that from the internodal segments of tobacco stems, the callus, the mass of undifferentials, proliferated only from the tobacco stem. If you take some of mass of these cells, proliferated only if, in addition to oxygen plus the nutrient medium was supplemented with one of the following extracts of vascular tissue. Vascular tissue means xylem phloem, yeast extract, coconut milk, or DNA. If these two oxygens plus any of these are mixed and put on this callus, the callus proliferate. Coog and Miller later identified and crystallized the cytokinase promoting active substance that they termed kinetin. When they just mixed this oxygen plus this particular any one of these and put on this callus of the tobacco, they could see that callus of tobacco can proliferate. It started dividing and started producing more and more cells. Now they wanted to find out what is there in the cells which started dividing. Skoog and Miller later identified and crystallized the cytokinesis promoting substance. Cytokinesis promoting substance in the mass, which promotes the cell division. They started to know what is there in that cells which started dividing. They just identified that and they termed that particular substance which is responsible for cell division as a kinetin. So that means these two together bring about the production of kinetin. Kinetin is cell division promoting hormone. cell division promoting a phytohormone. During mid-1960s, three independent research by three researchers reported the purification and chemical characterization of three different kinds of inhibitors. Three researchers. Three researchers resulted in three different kinds of inhibitors, inhibitor B, abscissation 2, and dormine. These are the three different types of chemicals or inhibitors produced by three independent researchers. Later, all the three were proved to be chemically identical. Of course, they are not knowing. When X was doing, Y was not knowing. When Y was doing, Z was knowing. These three were carrying out independent researches, and they came out to some chemical substances, some sort of inhibitors, and they called inhibitor B, abscission 2, and dormine. Later, all the three were proved to be chemically identical. It was named abscisic acid. All these three are called by one name, abscisic acid, ABA. Cousins confirmed the release. Cousin is a scientist. Confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored unripened bananas. What he said, what he found, cousin confirmed the release of volatile substance, some substance, which got released from the ripened oranges that fastened or hastened the ripening of stored unripened bananas. What they did? They took some substances from the ripened oranges, put these substances in a container having unripe bananas, and it resulted getting ripened. Later, this volatile substance was identified as ethylene, a gaseous plant growth regulator. Let us study some of the physiological effects of these five categories of features in the next part. Now, so this we shall, right, of course, this we are going to take up in a separate section, that is all the plant growth regulators we are going to study one by one. So, in nutshell, what all we have studied today, let us just go to that. I shall brief that. I must say, this is one of the most important part. Generally, the students find it very difficult. Prior to that, let me make it very clear what is differentiation, de-differentiation, and redifferentiation. So, this particular slide holds good. You know, there is what is called root apical meristem, shoot apical meristem. You also know there is what is called cambium, a patch of tissue, which has the ability to divide, found between the xylem and the phloem of the vascular bundle. What type of vascular bundle? Radial vascular bundles. This root apical meristem, shoot apical meristem, and cambium, they only have the capacity to divide. And root apical, shoot apical meristem, cambium differentiate in the sense they start dividing and forming some of the cells. And these cells structurally they get modified and start doing some function. This is what we call differentiation. What is the product of differentiation? Cell wall and protoplasm. In these, whatever the differentiated cells are there, they undergo changes. Differentiated tracheal element cells lose their protoplasm. Cell wall and protoplasm undergoes changes, where you have seen the cell wall and the protoplasm to undergo changes in xylem elements. They are also undergo changes in that of, see, here, tracheal elements, tracheal vessels. Xylem elements include xylem vessel and tracheid. So here, they undergo changes. They undergo modification. In order to perform a function, they lose the protoplasm. When they lose the protoplasm, there will be continuous movement of the water with the minerals. In fact, in the xylem vessels, there is to be some sort of the plate. But this plate, they are nothing but the wall of the each cell. This plate shows perforation. Urochemicals perforation plates in the vessel elements in order to allow the water to move continuously. The same thing that happens here, the cytoplasm gets lost here, and so that you can see the water can directly move, right? So this is what the changes you observe. Cell wall and protoplasm undergoes changes. Differentiated track elements cells lose their protoplasm, develop a very strong elastic lignocellulosic secondary cell wall. I said lignocellulosic secondary cell wall, it makes these vessels more strong so that water can flow continuously to any height. So these are the product of differentiation. Once these cells got differentiated to perform a different function with modification structure, they lose the ability to divide. Okay, further, over. But under certain condition, these regain the power to divide. That is de-differentiation. They regain the power to divide. They form some other structures, undergo modification, and take up a function. Once these de-differentiated structures attain maturity, again they lose the ability to divide. And such structures again get the ability to divide. That is the redifferentiation. So, development of interfascicular cambium, cork cambium, from the fully differentiated parenchymal cells is the example of de-differentiation. What is the de-differentiation? The living differentiated cells, these living differentiated cells that by now which have lost the capacity to divide can regain the capacity of division under certain condition, and it is called de-differentiation. And de-differentiation results in structures like formation of interfascicular cambium and cork cambium. And this development of interfascicular cambium and cork cambium from the fully differentiated parenchymal cells is the example of de-differentiation. Cells produced by the de-differentiated cells again lose the capacity to divide and mature to perform specific function, and it's called redifferentiation. So, I have told you, I have mentioned about that. This is what meristem is the structure, cambium is the structure, trachea is the structure, and interfascicular cambium is to bring about secondary growth in the dicot stem and root. We have seen that as a result of de-differentiation, you come across interfascicular cambium to form. You come across cork cambium to get formed. And this interfascicular cambium is to bring about second growth in the dicot stem and root. Cork cambium is a tissue found in many vascular plants as a part of epidermis. Cork cambium is a lateral meristem. It is found at the end, at the outermost periphery, and is responsible for second growth that replaces the epidermis in the roots and stems. And it leads cork cambium is always found in the woody and herbaceous dicot, gymnosperms, and some monocots. That you have already studied in the chapter 6. Okay? Now, this what I wanted to tell you about the development you already studied, what is the plasmatic growth you have studied, and this I have said that this is what is called plasticity. Plasticity is the phenomenon where different kinds of structures develop in response to the environment in certain plants. And the best example, this ability is called plasticity, and plasticity best example is heterophily. That means two types of different appearance of a leaf. Heterophily in cotton, coriander, and larkspur. This is what the examples have. Larkspur and buttercup. I already told you about the coriander leaves which we are using in our day-to-day life. Okay, this is the same thing. Yes, over. Then also I have mentioned about the growth and the development by that of the growth regulators. We have seen that there are these are the four important chemicals, namely that of the indole compounds, adenine derivatives, carotenoid derivatives, and gases. This is very, very important to remember. Which are the indole compounds? Indole 3-acetic acid, IAA, which are the adenine derivatives, N6 perfuryl aminopurine and carotene. And the carotenoids. These you have to buy it. There is no other go. There's no other go. Right? And as I've told you, these growth regulators are also called as growth substances, plant hormones or phytohormones. And these plant growth regulators are of two types. One is plant growth promoters, auxins, gibberellins, and cytokinins, and another is growth inhibitors, which mainly include abscisic acid and ethylene. Right? These are the growth inhibitors. And the growth regulators or inhibitors, they are all got discovered by accident. No one has done any specific effort to find out any particular growth regulator. And you must know these following scientists who are responsible for that. Auxin was isolated by the work of F.W. and this also. You must know who got which particular plant hormone or growth regulators. And Kurosawa was responsible for gibberellic acid, and Skoog and Miller, they were responsible for carnitine, and you have cousins who are responsible for finding out what particular substance act as the growth inhibitor, that is ethylene, ethylene. And also you must know that it was named as the abscisic acid because of these three different researchers which resulted in the formation of this abscisic acid. So with this, we shall conclude today's class and we shall continue next class with the physiological effects of growth regulators. Right?
Welcome to Sushruta app. In this video we are going to demo NEET topics and its related resources from slides and videos. Kindly give all the necessary permission and proceed to the main screen. Let us select Biology 11th as our subject. This will open up the syllabus for this subject. All the syllabus data regarding the subject will be loaded at this stage. Once the process is complete, the icon will be active. Zoom a bit so that the topics are clearly visible and click on the orange highlighter button. This will open up a selection box. Adjust the box so that it covers the topic you are interested in. Once this is done, confirm your selection by clicking on the green button. This will download the slides resources for that unit. Once the download is complete, it will ask you for the permission to download the textbooks from the NCERT site. Once the download is complete, it will open up the chapter and the subchapter hierarchy for the unit. Select one of the subchapter. In this case I am selecting the first subchapter. It will load all the resources pertaining to that topic. Let us click the second icon, that is textbook. You will be given an option if there are many resources. Now you can see, it has directly taken us to the page of the selected topic. Click on the small green button to load all the icons back. Similarly, let us select the third icon, that is slides. It will open up the slides for the said topic. Finally, let us select the third icon, that is YouTube videos. This will show the videos related to the topic from IIT-PAL. If there are more than one video, select any one of the links and watch the video. Similarly, let us select another topic and follow the same procedure. Remember, downloading of slides is a one-time process per unit. Slides are being downloaded here, as we have visited this unit for the first time. The next time we visit the same unit, slides will be retrieved locally. We can also select a topic from slides and get its equivalent matching resource. In this case, I am selecting the topic, taxonomic categories, and getting its equivalent topic from the textbook. So here we have the textbook resource showing taxonomic categories. When there are too many topics selected, or the topic name match is not found, the list of entire topics will be shown to the student. He can select any one topic and proceed further. Let's click on the Moodle icon, seen on the syllabus screen. This will take you to the Moodle calendar, where you will be able to view all the scheduled events. Let us click on one of the events. There is an event for Biology Unit 1. On clicking it, will give a live lecture Google Meet link in it. Clicking on the link will directly open up Google Meet app where the lecture will be taken. Let us select a topic from the biology syllabus and proceed further. Now let us click the first icon, that is Gyan Chat. Gyan Chat is a medium where the students can interact with the other students via channels. Each subject unit will be having its own dedicated channel. Students can share their doubts or thoughts via this channel. Gyan Moodle would directly take you to the course material for that particular unit. These course materials will be uploaded on Moodle by the lectures. Now let us try out a different subject, that is physics. Let us click the textbook icon and select the second option. This will open up a message box displaying the page number of the Kindle version of H.C. Verma book. On clicking OK, it will open up the Kindle app with the book loaded. Once the book is loaded, click on the hamburger icon on the top and select the Go To option. This opens up a dialog box asking you to enter the page number. Enter the same page number, which was shown to you earlier, and click OK. This will directly take you to the page related to the topic you selected. Now let us see the formula sheet for physics. Formula sheet is provided to get a quick reference of all the formulas covered in the syllabus. If you want to refer to a topic from the formula sheet, simply select the topic and proceed further. we are also providing a link to the CEngage app. On clicking the CEngage icon, it will open the CEngage app. You will find all the test material inside My Book Self option. Finally, you can take the mock tests using the National Test Apical app. Clicking on the icon will take you directly to the National Test Apical app, where you can try out mock tests. Whenever a QR code is found inside the textbook, the student can select the QR code and he will be provided with e-content material. In this particular case, we have contents related to text PDF. In some cases, there will be video content, graphic and interactive content. Let us see how video content would look like from a different chapter.
Hello my dear students, very glad to meet you all. I am Prof G.K Bhat. In 2018 or during 2018 to 2020, I got an opportunity to visit nearly 25 institutions. And I used to teach in all these institutions biology, preparing the students for NEET exam. For NEET exam, you must be thorough with each and every word given in NCERT book. That means biology, you have to by heart NCERT book. Not simply by hearting, understanding the concept and by hearting. That is what is expected. Now let us start with this particular chapter, that is Animal Kingdom, and we shall look into the various components that is given here in the Animal Kingdom chapter. Under basis of classification, what we are looking, what characters, what points we are looking to classify the animals. That is what we are studying under basis of classification. In spite of differences in structure and form of different animals, there are a lot of difference in the structure, in their form, gross morphology. There are certain fundamental features common to various individuals. There are certain features common to various individuals in relation to arrangement of cells, body symmetry, nature of coelom, patterns of digestive system, circulatory system, or respiratory.
We also have live lectures by world-class and dedicated tutors.
Well, that's all for this demo. Thanks for watching.